Good morning, everyone. Thank you for coming here today. Thank you for being here. I'd like to present my thesis discussion that I presented in June of last year. Thank you. So, without further ado, this is my master's thesis on the short-term effect of the megafire in Sierra de la Culebra on soil fungal communities. So wildfires are objectively one of the worst global disasters we must face, and they're the major disruptors of forest ecosystems worldwide. And the ongoing effects of climate change are only worsening the frequency and intensity of these cataclysms. So much so that so-called megafires, which are the fires that affect more than 45,000 hectares, they're spreading throughout the planet at an unprecedented speed and alarming rate. The Sierra de la Culebra megafire in question occurred near Zamora in Spain in the summer of 2022. It was a series of 3 closely occurring fires which sparked by lightning in June, and for 2 whole months, firefighters struggled to put out the flames. Over 75,000 hectares were affected. Homes were destroyed, and the duration and intensity was never seen before in Spain. And this was exacerbated by the hottest and driest spring on record in the peninsula's history. So, speaking of fungi, they're thought to be about 5 million species, but I'm going to talk about specifically the 2 biggest groups, saprotrophs and ectomycorrhizal fungi. These are the 2 major types that we can find in nature in many shapes and forms, and what you see here above ground is the carpophore. Specifically, we're evaluating the impact of soil fungi, so we may not always see them, since thousands of species are microscopic organisms that may only surface in a mold-like form. So these soil fungi provide fundamental ecosystem services such as nutrient cycling, decomposition, enhancing plant productivity, and water uptake. Saprotrophs generally facilitate decomposition processes of dead plant matter, returning nutrients to the soil. And ectomycorrhizal fungi, on the other hand, like to keep plants alive by creating mycorrhizal associations with plants and tree roots. This helps the plant uptake nutrients in exchange for carbon. So ECM, ectomycorrhizal fungi, are generally the ones that we consume as food, although there are several saprotrophic species that are edible. So what are we looking to answer by analyzing the effect of this megafire on soil fungi? So it's fundamental that we analyze these key organisms in the proper functioning of forest ecosystems, but not only forest ecosystems. So we looked at answering these 4 questions in particular. The effect and richness on the richness and diversity of fungal communities, the effect on the composition and turnover, the effect on pyrophilous fungi, and the effect of the megafire on edible fungi. So how do we do this? The area of study is located in the northwest of Spain, near Zamora, at the elevation about 850 meters above sea level. We analyzed 3 separate blocks of Pinus pinaster plantations, which presented both burned and unburned situations adjacent to one another. The understory of these 3 areas were mainly saplings of Quercus ilex, Quercus pyrenaica, and as well as plentiful Cistus ladanifer and Erica australis. We subdivided the block into 6 plots, within which we created 3 burned and 3 unburned segments of 2 by 50 meters. Within each segment, we extracted soil cores from the first 10 centimeters of the profile, avoiding contact with nearby plants. Afterwards, we took the cores back to the lab, where we sifted the soil to remove larger particulates. Then we sent the samples to IDE Forest, where the DNA extraction was performed according to standard PCR procedures. We performed the metagenomic sampling, which through the Unite Fungal ITS database displayed operational taxonomic units, the OTUs. Then the data was transferred to RStudio. An ANOVA test was performed, the permutational multivariate analysis variance, PermANOVA, non-metric multidimensional scaling, and finally a multi-level pattern analysis was performed to assign taxa that were significantly associated to each treatment. The results of the combination of lab and computer software analysis was in line with what we expected. The fire had decimated the Basidiomycota, which were associated with the burnt trees, and the Ascomycota flourished, which are the phyla associated with saprotrophs, the decomposers. The remaining phyla are difficult to analyze, especially since only 2 were significantly affected by the fire. Here we can see that the saprotrophs dominate, while ectomycorrhizal have been severely reduced in quantity. According to the results, the effect of the fire on fungal richness was not significant, although the Shannon Diversity Index produced a highly significant shift, similarly to the evenness result. So these are the major phyla affected by the fire. You can see Basidiomycota and Ascomycota are the major ones. So let's analyze the possible causes and connotations to our research question answers. In the experiment plots, an overall visible devastation to the area was witnessed, especially when comparing to the adjacent control areas. Richness and abundance decreased, as expected, and certain gills and phylum were seen either resisting to the heat of the wildfire, directly proliferating, thanks to the heat, or colonizing the new habitats and niches that were left behind by the fire. Continuous and appropriate forest management becomes an indispensable practice to avert deforestation and the loss of ecosystem due to wildfires, which is the direct cause to the decrease of these 2 indices. The impact on fungal communities was evident from the analysis of the sample in the study. As I mentioned previously, ECM were eradicated by the fire, and the surviving hyphae perished since they were unable to locate new hosts. Oh, that's a great picture. Yeah, so the results, in conclusion, saprotrophic fungi, on the other hand, as opposed to the ectomycorrhizal, have better adapted themselves to fire conditions by evolving heat-tolerant or heat-resistant mycelia. This is convenient since this group is able to feed off of the nutrients left behind by the fire. Since many fungi change their lifestyle behavior, some pathogenic fungi are also capable of becoming saprotrophs, which could explain the large spike in saprotrophs post-fire. So pyrophilous fungi are a very interesting group. Most species can be observed shortly after the fire, sometimes within a matter of days. And the reason behind their appearance is their ability to adapt to fire by producing anatomical structures such as large fire heat-resistant or heat-induced sclerotia, pseudosclerotia, conidia, and dormant spores. The burn plot showed that Ascomycota is the leading phylum, and this group contains many fungi associated with the Pisizalis orders, such as Pisiza pyronema, as the name suggests. And then contrary to the behavior of most ectomycorrhizal fungi, results show that a select number of Basidiomycota actually have proliferated in the post-fire conditions, namely Foliota, Leophyllum, and Terfetzia. One of the OTUs with the highest positive impact post-fire, therefore an indicator species, was identified belonging to the Coniotherium genus. Fungi within this group have been determined as agents in combating plant pathogens, agencies such as Bayer use them. Since this species is mainly applied in the agricultural sector, further research is needed to determine its role and potential application within the forest ecosystem for ecological restoration purposes. So further studies are needed, obviously, to ascertain the key drivers for this rapid post-fire multiplication of pyrophilous fungi, whether it's due to their direct adaptations to fire or whether the biotic and abiotic changes in its habitat allow it to proliferate. Either way, it's evident that the forest's ecological processes are prepared to host a destructive event, such as a devastating megafire. So by keeping heat-resistant spore banks underground. And as far as natural resources such as the edible fungi, in the last 5 decades, the consumption for edible fungi globally has increased over 20-fold. So demand for wild ectomycorrhizal edible mushrooms in Spain has not been an exception to this trend. consisting with findings related to the decline of ectomycorrhizal fungi. A strong decline was observed in the edible fungi in the sites of the study. So the vast majority of these were found within the Basidiomycota phylum, since the ectomycorrhizal associations with the forest flora. So in conclusion, this research provides pioneer results in the effect of a megafire on fungal communities in the Mediterranean region, which could provide useful evidence-based information for the adequate restoration of affected areas within. Thank you so much.